Thanks very much. I'm as confused as ever, but it's that's maybe just me. Our next paper, Jonathan Etherington of the MBFI, going to tell us a bit about application.
Thank you. Good morning, ladies and gentlemen. It's an honor for me to stand up here this morning and be able to have a couple of minutes of your time. We're going to start about Rhizobia technologies. The speakers before, and I think each one of us in the room really look at sustainability, look at the furthering of technologies, but also being able to be conscious about our world environment or our environment, our human impact. And our first slide for today is actually looking at that, sustainability. So if we were to look at soybean inoculants and amounts of nitrogen that could fix compared to utilizing oil or burning oil to produce chemical nitrogen fertilizer, we made a theoretical sum. Excuse me. Sorry. So for that, we have to burn 479 million liters of oil to produce 362,000 tons of urea in nitrogen, for nitrogen. So that will mean that 11,322 trucks or links of fertilizer or nitrogen fertilizer would have to leave our premises to produce that amount of nitrogen. In Rhizobia technology, if you had to make that conversion, only 5.2 truckloads or links would have to leave the same premises by utilizing only 160,000 liters of biologicals. That's a sustainable number that we can be able to utilize and be able to be proud of what our South African farmers, us as an industry, have been able to achieve. Okay, so what about the nitrogen and the importance of in plants? So first and foremost, pods require protein production, and this is a really important part of the nitrogen cycle. Plant tissue is another side of it. Plant growth and development. Leaves also need a high amount of nitrogen in chlorophyll production, and then roots need to have nutrients and water uptake or usage. So nitrogen is important in all types of the plant, or all sides of the plant, and it's a really important component in soybean management, or legume management as a whole. So let's look at some important points of nitrogen in the plant. Soybeans produce or can utilize between 70 and 105 kilograms of nitrogen per ton of soybeans. We know from a scientific fact that 94% of the yield is directly linked to nitrogen assimilation in soybeans. Nitrogen assimilation, is directly linked to soybean increases, and from R1 stage in the plant up into R5, reproductive stage 1 to reproductive stage 5, up to 4.5 kilograms of nitrogen is required per day to fill those pods and increase yield at the end of the day. So carbon to nitrogen ratio in the soils influences nodulation and nitrogen fixation process. And this is an interesting fact in our country compared to different countries around the world. Our carbon to nitrogen ratio is very different to various parts of the world. Synthetic nitrogen sources involve high oil demands and create high pollution effects. We need to be able to produce 60% more protein for the world's population with 80% less emissions. And through nitrogen fixation, we can definitely achieve a portion of this. So the importance of nitrogen in plants. If we look at soybeans, we have different species of Rhizobia that could be utilized, but it can produce between 180 and 250 kilograms of nitrogen per hectare. When we do regulatory trials that Televali was talking about, we actually utilize 200 kgs of nitrogen compared to inoculants. So that's pure nitrogen when we do as a treatment or a chemical check to make sure that, that we can prove that the Rhizobia is actually giving enough nitrogen and fixing enough nitrogen for the crop. If we look at all the different, let's say, legume crops, you can see peanuts 100 to 120 kgs of nitrogen, lupins 150 kgs, alfalfa, lucerne up to 250 kgs, and various different legumes use different amounts of nitrogen per hectare, or can be or fixed by Rhizobia. So Rhizobium bacteria, it's a gram-negative bacteria. It's also a mobile one, and we also talk about it as a medium in size. I'm not going to go into all the technical side of Rhizobia, but looking at more the infection process and how we actually how we could utilize it. So if we had to look at the infection process, first we start off with the recognition of the Rhizobium with the plant, or the interaction. The next one is the penetration, the infection, and the growing of the thread. The bacteroid is actually then transformed, and then the nodule begins to form. So if we had to look at 7 different processes in this, so first and foremost, just like humans, just like as far as our interaction is concerned, we need to be able to, first and foremost, the root creates flavonoids, which then bind to the plasma membrane of the Rhizobium, and that creates knot factors. So there's a communication between each other. And it's very specific between legume and Rhizobia that you're actually going to be able to connect those knot factors, or the production of knot factors and flavonoids. While the receptor is binding, or that causes an increase in calcium in the hair root, which also then causes the swelling of their tips and the curling around of the Rhizobia. Rhizobium is then injected in proteins to induce root development and infection of threads, or infection threads. These proteins are known as nodulins, and they actually they create cell division, and then the formation of a nodule is formed. Nodules also are pink inside, and they are regulated by leg hemoglobin, which we'll get into a bit more detail. Nodules are developed in vascular tissue and transports nitrogen components to the shoots, organic carbon form to the other parts of the bacteroids. Okay, so let's talk about communication. So first, that interaction is really important. So if I had to say, good morning, sir. My name is Jonathan.
Good morning.
I'm
Rolf.
Hi, Rolf. Good to meet you. Exactly the same as the communication of a soybean inoculant with a soybean plant. That interaction is very, very important. So one is created as a flavonoid, the second one is a knot factor. And if we be able to communicate with each other, we can then be able to find a synergy between each other. This attachment of soybean roots or soybeans happened really, really quickly. And it can happen within a 5 minutes time frame. So in acidic soils, which is generally a major situation in South Africa, but we also do have alkaline soils down in the Northern Cape, different types of proteins are produced from the bacteria. First and foremost, lectins in the acid soil and other proteins in the alkaline soil. And this will help with the receptor and the communication between the Bradyrhizobium and the soybean itself. So if we look at this is an infection. So this is, if you look at first slide over here, that's where the bacteria, this is a stained marker. If we look at on the side there on C, it says 2 days or 2 DPI, days post-infection or interaction. And you can actually see how the curling has happened. The bacteria is starting to multiply. 3 days afterwards, you'll see that multiplication of bacteria, and 4 days after that. So this happens really, really quickly. And we need to be conscious of that speed, that effect that it actually has. The next thing in importance is the calcium portion in the roots. So as the attachment forms on the root hair, an increase in calcium influx happens, and so the curling of the roots happens around the bacteria. This is really important as far as to increase from the knot factor but also increasing the calcium in that specific region. And as that pointing of where the bacteria is attached, let's say it's at the bottom here, you'll see that there's an increase in calcium level, and this root here will then be able to curl around that nodule of the bacteria and actually infect and make a nodule. Second one is the infection thread. So this producing, once it's actually, the recognition has happened, there's actually an attachment, a root hair curling, then there's something that's called infection thread, where the bacteria actually infects and goes into the plant. Second thing, in the rhizobium moves through the infection thread and penetrates the root cortex. In absence of boron, these glycoproteins also have an ability to be or not progress as quickly as they would like. Second is the division of the root hairs, and this is really important, which Adriana was talking about, cobalt, molybdenum, and then other cofactors like copper, for argument's sake. Really important cofactor as far as the affinity of the actual nodule is. If we look at the next post or next slide, you'll see that this is the actual vascular tissue over here, and this is sitting at the cortex of your root, and you're actually seeing where the nitrogenase, or first of all the bacteria growing, and this is where the nodule is creating the nitrogenase, pushing it in, or nitrogen, and pushing it back into the plant. So it's a really integral part of the plant, but it's also not, if I may ask, what is a nodule? Is it a bacteria or is it a plant? Both, yeah. So it's a bacteroid. It's something very, very different to just being one, a bacteria starting off as a bacterium plant, but they become one, becoming a bacteroid. And that's a very important thing as the whole change in because it's a symbiotic relationship. Conversion of N2 to ammonia. So this is one of the most obviously important parts of it. Atmospheric nitrogen is not reactive as a general N2 gas. Rhizobium then takes that N2 gas and it makes it ammonia. So we're utilizing the 78% of our atmosphere's compound. And this conversion requires enzymes known as nitrogenase. Most nitrogenase are inhibited by oxygen. So this is quite an interesting point on the Brady rhizobium, is that most nitrogenases are inhibited by oxygen. So we have something called, or the nodule has something called, or the bacteroid has something called leg hemoglobin. Leg hemoglobin binds to this oxygen and also then controls the oxygen amount within the nodule so that you can actually create this nitrogenase or ammonia, which is then available to the plant. If you look at molybdenum and iron, in the conversion of N2 gas and ammonia, you can see that directly linked, these coenzymes or nutrients are part of a coenzyme in the nitrogenase activity, and that sits right in the middle. And with the absence of this, you do get a reduction in nitrogenase activity. And it's a really important factor in all crop or in all soybean production around the world. One thing we do have, and something that we are very different to a lot of countries in the world, we have quite an acid pH. So iron is very available in our soils. So generally iron utilization in South Africa compared to other parts of the world would not be required. Compounds like cobalt, molybdenum, absolutely a major importance in our pH of our soil. So in this importance conversion of N2 gas to ammonia, leg hemoglobin is an abundant protein, and we need to be able to utilize this or iron to create hemic iron, which also then buffers the oxygen content in the bacteroid. Then on nodulation or nodule senescence. So if we see over here, the first, right at the top there, 4 weeks, we have a pink interior, and that's the presence of high leg hemoglobin. If you look at number 14 weeks later, the interior becomes green from the center outwards, and then at 18 weeks, generally the symbiosis has ended, and that interior becomes brown. So the importance of this nitrogen the iron, and also then other cofactors in the enzymes. Conversion factor is really important to keep that stimulation as long as possible. So what is important about inoculant and what important features are there? So if we look at laboratory control measures, really important from a petri dish all the way through laboratory QC quality controls. Then also having grain chambers, so batch testing of every product to actually see that it's producing nodules in the correct amount, in the correct position, and also then the volume of, or the activity of the bacteria. Then last but not least is then actually doing in-field work around the country in different climatic zones. If we look at this, in the very sandy soils conditions, in Delmas, very high clay soils, totally different situation and totally different microclimate. It's important to test all the products in all the different climatic zones. So, nitrogen fixation products. Okay, I have a little bit of a question. So let's look at primary and secondary root nodules. If we look at primary root nodules, you'll see that they're sitting right on their crown. We normally talk about a matchbox around about the soil level compared to secondary root nodules. Could anyone help me to understand what is the difference between, for argument's sake, if we had over here on the secondary root nodule, we had 1 gram of dry nodular mass compared to 1 gram over here. What would be the difference in the nitrogen fixation percentage, or in multiplication? What would be the difference? So if we had 1 gram over here on a secondary root nodule and 1 gram over here, so identical amounts of nodules formed on different parts of the root structure, what type of difference in nitrogenase or nitrogen activity would there be? Sorry? So 100%. So 10 times difference in nitrogen fixation if you're in a primary nodule, the same weight, not the size, same weight, same dry mass. Over here, you'll fix 10 times more nitrogen for a nodule sitting over there compared to that on the secondary root over there. So position is really important on a nitrogen fixation. So here's just a clip as far as what the leg hemoglobin looks like. Adriana did talk about something like a bioinducer, new technologies coming along, and you'll be able to see that this redness of the nodule actually increases with different technologies as we increase the communication between the plant and the actual rhizobia, making newer technologies come forward and making more nitrogen fixation. Over here, you'll see brown nodules. These are dead. They've stopped their senescence. They're actually not available to the plant. And in tough conditions, in very high drought conditions, when the plant is struggling to get water, it can also let those nodules go and then be able to, at the end of the day, they will actually let them go because they need to protect themselves. So if we look at, for argument's sake, concentration of inoculant, if here we've got a slide where we started off with obviously a very low number, or 0, 10 to the power of 7, 10 to the power of 8, 10 to the power of 9, and 10 to the power of 10. So these individual products over here, there's 10 times concentration difference. Again, another 10 times jump, and a 10 times jump. So from there, 10, 100, 1,000 times jump. So it's very different to a biological or an active ingredient, which is generally, if you have 100 grams per kilogram, compared to a 500 gram, there's 5 times. This is now 1,000 times difference in concentration. Okay, so when reading a label, it's really important to look at that concentration level and the effectiveness of that concentration level. So if we had to look at the dark green bars, the bottom over here, you'll see that this is the primary root nodes or nodules that are forming on the primary root. On the top of the light green over here, you'll see this is the secondary nodules formed from the inoculant. So as the concentration increases of the inoculant, we're finding that we have more nodules formed on the primary root structure compared to the secondary root structure. But even though that we can see over here this looks quite similar. It's actually a secondary root nodules that are increasing, not the primary root nodules. And we went back and said, or if we went back a couple of slides, we said that there would be 10 times difference in nitrogen fixation or nitrogen simulation in the plant if you had a nodule that was on the primary root. And that is really important. Here, by just increasing the concentration from 10 to the power of 9 to 10 to the power of 10 is more than, or close to double of nitrogen activity. Okay. So other factors on how bacterial products work. Bacteria nodules facilitate a lot of different or nitrogen-based compounds. They also have symbiosis, and this means that there's a mutual relationship between the soybean plant and also the bacteria. So if we had to look at, for argument's sake, variations of seed treatments that we would like to be able to look at or give or put on the seed coating, we would classify this as being from very good on every seed, 5 times 10 to the power of 5 bacteria or Rhizobia per seed, compared to a very bad treatment of less than 1 times 10 to the 4 per seed. This is very important as far as industrialization is concerned, but also with mechanization of products or treatments in the field. We need to make sure that the quality of our treatment is always sitting around about that figure over there. With new technologies, high concentrations, definitely going to be able to meet this number fairly confidently on about 95% of the time. Second thing, or on factors of contributing to high quality inoculants, first of all, it must be registered, as Tilavali said, under Act 36 of 1947. If we look at older concentration or liquid products with a premium, let's say Rhizobium isolate at 2 times 10 to the power of 9, this was the old regulation. That would be a minimum for a liquid product to be a soybean inoculant or registered by the Department of Agriculture. New technologies with variation of different products and new fermentation procedures, we can see that new products are getting to 2 times 10 to the power of 9, which is 10 times more than traditional older technologies in liquid fermentation. Second, a couple of other things: no contamination, strict internal qualities, ensuring that shelf life is really, really important. Some of the older products that we started out as right at the beginning had shelf lives of 3 months. Today, I can stand here, and we've gone through this with 18-month shelf lives. And this makes sure that the farmer that's buying the product this year, not only can he use it for this year or immediately, but can actually utilize it for next year if he can store it properly and is actually correctly utilizing the product. So adequate shelf life is a major, major importance. Next one, packaging material, on-seed survival. A huge, huge debate, but for another day, because there's a lot of factors that we can chat about on seed survival. Compatibility and additives to improve the seed and soil survival go all hand in hand. If we look at dry and wet applications, this has been a talking point for a long time. How do we look at different applications? So dry application of inoculant or wet? So if we had to look at, on the first and foremost, So if we had to look at dry, we get generally a low seed coverage, a low control of the application, less effective because of the dispersion or the application on the seed itself, and then it's a manual application in both production facilities but also in field use. So it's quite a difficult product to handle, and definitely from the origins of Rhizobium production from the 1970s, 1980s, definitely this is how we started in the industry. If we look at wet applications or liquid fermentation, better coverage of the seed, control of your application quality, more efficient from the product use side of it, and also then being able to automate or be able to precisionally put on the seed through different treaters or seed treatment machines. So ideals or properties for pre-treatment survival. And as farmers, as industry, we want dry, plantable seed as best as possible, and especially in high humidities, when the rain is coming, that plant has stood outside, we actually want this real plantable, unsticky seed. Unfortunately, bacteria like exactly the opposite. They like the sticky, wet, completely moist condition to survive. So how do we balance the two out? So first and foremost, we want high viable bacteria and fungi with extended storage periods. We want good distribution of the product on the seed. We want low stickiness, or at least good flowability. The ease of the treatment, increased compatibility with different products, chemical components, biological components, and so then also be able to have additional drying products that you can apply to the seed to reduce the stickiness of the seed. But the biggest problem, again, is that balance between stickiness, bacteria world, and dry plantability for practical world. So desiccation is, however, the most important factor for poor survival on the seed. If we look at just, for argument's sake, several technologies that were developed, here we have on the left-hand side, here you can see this is a bare seed over here. You can see it's fairly smooth, but if you're looking over there, that is a bacteria, a Rhizobia. If we look at it, if we had to apply, so this bacteria is now has all the factors against it to dry out. It's got this dry seed, moisture of 8%, specifically in this country. If you look in Europe or in Brazil and Argentina and different parts of Latin and North America, we're looking at 12 to 14% or 12% seed conditions. But on the condition as far as the seed is here, very, very dry. With a protector, you can see the bacteria sitting there in a protected area. But this changes the surface of the seed, makes it a bit more lumpy. We can't maybe see that, but actually increases or changes the surface of the seed. But this will also then protect the bacteria from desiccation. Second technology would be osmoprotection. And osmoprotection is basically being able to keep available the water within the cell of the bacteria for as long as possible. So if we had to look at, for argument's sake, with osmoprotection, you'll be able to retain the water as long as possible. Without osmoprotection in a broth media, which is what Adriana showed a bit as well, is the increasing of the H2O leaking from the cell or from the membrane. I think most importantly, I've got only two more slides, is actually after-sales service and after-sales evaluation of your nodules, of the practice of what you've done in the field. So we've split the country in two divisions, one eastern section, so basically the N1 from Johannesburg to Bloemfontein, everything east of that, and anything west of that. So first, let's look at the eastern side. West is almost identical. It just has a bit more days because of temperature and also sandy conditions. So if we had to do evaluations from on the eastern side, 40 days after planting or after emergence would be the best time to visit that soybean field as far as nodulation is concerned. That's when we would evaluate where the nodules are, how well they've done, and also what the treatment looked like. Then, if there's good nodulation, that's perfect. We can then do a follow recommendation of phosphate, potassium, molybdenum to increase that nitrogen or cobalt, manganese, depending on what formulations or what crop protection program you're using. But then you can recommend different variations to increase that enzyme activity or coenzyme factors. If there's not a sufficient nodules, and this is where we have to go, we'll have to go and recheck that field around about 60 days afterwards, 60 days after emergence. So that's about 20 days post that first visit. And then we would have to estimate again, if we had few nodulation or low nodulation, we need to understand a couple of concerns. First and foremost, we need to track that batch of product to see exactly the viability and traceability of it. Was it cold or extremely cold, wet, or extremely hot and dry at planting? This could also cause a major difference as far as, so we're going to have to start asking questions about what is actually happening. pH problems in the soil. Is this only a small patch? Is it localized, or is this throughout the bank? And we've got to be able to, or once we've established that, we can then be able to say, are the plants even flowering? Are they in R1 and R2 stages? If we can, let's say, answer all those questions and we can't rule out one of those things, we would then be able to come back 20 days later at around 80 days after emergence, and if there's still no nodulation in that field, we would then have to decide how we're going to approach using chemical nitrogen fertilizer, either by using LAN or urea. Would we do one application?